Okay. Now, if I ask you this question, I mean, how do you understand that concept? Number one, do you know why what is the reason that everybody different to each other? Why it is important that person to person are different to each other to have like this concept?
Sir, they are genetically different.
Genetically different. Okay. Why genetically different is important? Okay, maybe hard to explain, right, for you? Right. It's a very big concept to understand.
Sir, for evolution.
Right now, Apurva, you said mutation. Mutation can take as one cause for the difference, right? We have to find first why everybody become genetically different. What is the reason to become genetically different? Second one, why it is important. So we have two questions. So the two questions are, what is the reason to become genetically different and how it happened? Second one, why it is important, right? I will answer the second one as the, I think what Disat mentioned, the evolution. It's a, how do you understand evolution? Like, you can say something like, okay, evolution is this. Again, if you look at evolution definition, maybe the changes of the characteristics over period of time in a population. Changes of the characteristics in a population over period of time. I mean, change. That means the concept is the world, what we live here on this our earth, nature, how we evolved or how we born or how we formed here, make one very important things. It keep always differences. It keep always differences, and the differences are random. It's a very random differences. So these differences, every organism, they have differences in their body, in their function. Then what the nature thought, I don't know who is nature, what we call nature, like word use. The changes happen our surrounding. We are facing these changes always. Always we are, every day we are facing the changes around us. So then some organisms, they can face these changes because they are different. Others, they can't face these changes. So that's generally how it defined. Let's take a very simple example, which is not the always 100% best definition we can take. Now, virus or bacteria suddenly emerge somewhere, forest or in animals somewhere in the world. That bacteria or virus is the worst in the forever. Like we had, like it's a very most dangerous one. Like it happened in the 1300. There is a bacteria called Yersinia pestis. This bacteria suddenly pop up and spread among the population, and they spread many places in the world, and it killed 200 million people, like 1/3 of the world, maybe. Like we are close to 1/3 of the population killed. And we know that when the COVID-19 came to the world and simply said, okay, we don't have medicine. COVID-19, we don't have medicine. Vaccine is the only hope, so we try to make vaccine. We are developing technology. We are developing many ways. But at the moment, if very devastating pathogen virus or bacteria comes, if virus comes which can spread quickly and it can kill so many people, like maybe you heard about Ebola virus. Ebola virus is a very dangerous virus. It can kill like 70% of people affected. Like 100 people got this Ebola, only 30 will survive, 70 die. But luckily, Ebola spread not from the air. It is spread from the body contamination, so it's controlled. Now, likewise, we have so many examples when you're talking about the human only. I'm not talking about the plants and animal, because this world, plant, animal, all the organisms are important. Now, the things created the differences to face any kind of a challenge. Any kind of a challenge, as a human, we can face, but only some people, they can face it, they can survive. Others, they can't face. The general concept is like that, because you can understand that in the future, like another 10 years later, there is a very bad virus is coming. And how our body knows that virus is coming another 10 years, and we have to prepare our immune system to fight with that virus. Maybe something coming from the space, from aliens or whatever, some bad virus. And how our body knows that this strange virus can destroy by our immune system. Like it is all the possibilities creating. So we cannot do that. It's all the possibilities, chances, like everybody is different to each other. At least some will have the ability to fight with that virus. That is the general concept, right? So we will talk about in Unit 3 the evolution details, then we can understand. So significance of meiosis is the important thing is the genetic variation. So genetic variation is not an easy concept. The variation is difference among each other. Why it is different is important. We have to face the changes happening around us. At least some have the ability. Okay, then before we move to the the variation, how it happened, this variation, what is the reason for variation? Why everybody different to each other? We can list at least 5-6 causes, reasons we can list. As Apurva mentioned, the mutation is one. The mutation is, we will talk later on this thing, space DNA replication, there's a tiny mistake happen. Let's say example, there's a small mistake happen. At least one nucleotide change. Now out of this 6 billion nucleotides, just 1 or 2 nucleotide change. So these changes are not detectable by our the DNA repair system and DNA the screening. Like cell can check these errors. Cell has ability to check these errors. So always there are few changes happen, and these changes bring variations. But if we list all these causes for variation of genes, like genetic variation, what is the cause for genetic variation and why it is important? These are the two important questions. In biology, genetic variation, the strongest cause, the strongest factors are coming from meiosis. Strongest factors are coming from meiosis. And there are two factors which contribute for the variation. Remember, two factors. I think in meiosis, we have to learn only that too. So go through mitosis again, try to remember the steps. Then everything is same in meiosis. We don't want to, we should find the ways and methods and tricks to learn. Just not like a hard ways to learn is too much. So if you strongly get into your head, okay, two changes happen, that is important for genetic variation. Genetic variation is a big concept. It is a driving thing. The world is actually surviving because of the variation. So we talk about variation later on, because we have a new lessons about variation is coming up. But when you strongly getting the things to your head, you see the weightage. Variation is the very important concept. It's not light one, so you put effort too much to remember it. Okay, meiosis. There are two changes happen which you don't see in mitosis, and these two changes is important, right? So let's see these two changes. Let's learn these two changes. Now, in one happened in prophase, right? So what first one happened in a prophase, second one happened in a metaphase. Right? That means in meiosis, the prophase and metaphase is the important thing. The important prophase and metaphase. Again, there is a different thing than mitosis. We have to understand, right? It's very important to understand, and most of the time, it is not delivered properly to the student. What is the process? By heart, you can remember things, but it's not the way. You should understand things well. Then you can easily, you have an idea, it's clear in your mind, what is it, right? Okay, then I try to explain you like this. Now, mitosis, what I said, there are 46 chromosomes. That's a normal human cell has a 46 chromosomes. Okay, allow me to draw it like this, a chromosome. I draw chromosome like this. So that's a 46 of this we can see. And what happened? This duplicated now. DNA replication happened, and now it become like this. The yellow color one is the replicated one. Then each 46 chromosome, if this happened, then we have a 92, right? And then anaphase, what happened? It detached. That means one is separated like this, red color one, and the other color one, yellow color one separated. So they are exactly same. And 46 is going to this cell, 46 is going to this cell. Then the cells are genetically same. So that is the mitosis, right? So we understand in mitosis like this. Now when it comes to meiosis, okay, again, this is the chromosome 46. This chromosome duplicated to become 92. That's a duplicated one is a yellow color one, and this red color one is the one like this. Okay, then we try to see what is happening differently. To understand that, we need to come up with another important concept. If 23 coming from mother, 23 coming from father, 23 chromosomes and 23 chromosomes. And how this 46 chromosome exist in the cell. 23 coming from father, 23 coming from mother, and now 46. These 46 exist in pairs. So we call it pairs. So chromosome exist in pairs. And why they exist in pairs and how they pair up happen. What is the reason to pair up? Now, reason to pair up, 23 coming from mother and 23 coming from father, these chromosomes have a similarity. Are they same? No, they are not same. These 23 and 23, they are not exactly same. But they have similarities. They are, let's say they are very similar to each other. They are not 100% identical. They are not same, but they have similarities. Then what happened? This one is from, let's say, mother chromosome, and this one is from father chromosome. Now we can see this as a pair. And to this pair, we give a name. What is the name we given? Homologous. Now this word homologous, it says same. The general meaning of homologous is they are same. But I have to add homologous, but not identical, right? They are not identical. Not identical. So chromosomes, they are not identical. But they have a similarity. We call homologous chromosomes. Word use homologous chromosomes. I think I can, if I can show it like the, okay, here. Now here just picture of human chromosomes. They have these diagrams elsewhere and telling us, look, these are the human chromosomes, and human chromosome exists in 23 pairs. At least you can see 18 pairs here. And if we take one like this is one pair. Each one you can use the name, yes, they are homologous chromosome. Are you getting the word? Yeah, they are homologous chromosome. Number 6 homologous, 7 homologous, 8, 9, 10 pairs, they are homologous chromosome. But when you say homologous, you have to strongly keep in your mind, they are not identical. They are not identical. One coming from father, one coming from mother. So if we take a cell like this in the nucleus, we can see this is from one, and this is another one. They exist like this. Okay, now what will happen? So 46 become 92 here. So this one and this one is same to here. Now, when they form 92 chromosomes, what is differently happen now in meiosis? This pairing story, right? That means they try to stay as a pair in meiosis. Mitosis, you didn't see. If you remember the mitosis, the metaphase, there is a chromosome like this and attached to the spindle, and there's another one below it like this. Another one below it. So they line up like this. After they line up, so this side has a 46, this side has a 46. And then they break in the middle. This cell get 46, this cell get 46, genetically identical. They are genetically identical because the chromatids are same. Now here again also they make 92. So now I'm going to show you what is happening last, right? There is a gap here. I will show the gap, what is exactly happening. But when they separate in this meiosis, so I make a line here. Okay, now it's separated. So what happened, the metaphase or whatever happened, it's like this. Here is a 46, here is a 46. So chromosome number is 46 and 46, like here and here. Now what is different? No different. 46 number is 46, here number 46. Chromosome number in both case 46 and 46. Then why we call this case diploid and this case haploid? Diploid because there's a 46. If it is diploid, there should be 23. Now we don't see 23, we see 46 and 46. So where is the difference happen? Okay, different happen, we can understand like this. One thing is, this further continue. That means it's going again for the second division. So then that's a second division. So now meiosis has a second division. And then we can call this first division. So we can divide into division 1 and division 2. Division 1 and division 2. Now what is most important division, 1 or 2? Answer is division 1. Division 1 is the important. Okay, now meiosis is also called reduction division. Reduction means it's getting lower, something getting lower, reduced. So chromosome number reduced. But what is really happening at meiosis 1, right? So we can say meiosis 1, and this is meiosis 2. Actually, everything happened at the end of meiosis 1. Everything happened at the end of meiosis 1. We see number 46 and 46. It doesn't say anything to us by looking at the number, chromosome number. You need to remember it is not the number. It is the type of chromosomes going to each cells. It is a quality. It is a quality because now this 46, it make a division to make 23, 23, 23, 23. 46 divide again to make 2 cells. Chromosome number is 23. The second one also divide to make 2 cells, 23, 23. But now you see, okay, half number of chromosome 23. Now I see the difference is fine. They are 46, 23. But this number is 46 and 23 doesn't give a big picture to us because the difference of the chromosome, everything happened at meiosis 1. Right? So you need to remember this one. It happened at prophase 1 and it happened at metaphase 1. So if we learn prophase 1 and metaphase 1, other things are only mitosis. We don't want to again learn, it just we mentioned this. So then prophase 1, metaphase 1, anaphase 1, telophase 1. After that, prophase 2, metaphase 2, anaphase 2, telophase 2. Now I save you time. There are 8 stages. Out of the 8 stages, 6 stages you don't want to study. You only study mitosis. Only you have 2 new things to study here, meiosis. Only 2 things you have to study. Rest of the things, that mean anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. All you just park somewhere and I will show you how to handle these things. You focus only to learn these 2, prophase 1 and metaphase 1. And carefully look why the changes happen at these 2 stages. Why it is happened at these 2 stages. And also keep in mind the number 46 at the end of the meiosis 1, chromosome number is 46. But it goes to the second one, 23, 23 to make the number. But you see number it reduced, but that is not that important. Important is the variations, changes, where it happened. Okay, coming back, we continue to these changes, right? Now, homologous chromosome, they try to pair up now. That is the first thing going to happen. Like this, if this is the one chromosome, you can see this is duplicated one, right? So it is always like a mitosis, chromatid. This one is a sister chromatid. That's a duplicated one of this. And now the homologous one is coming up. Homologous is again like this. That's a homologous pair, and that's a duplicated one. Let's also try to understand 23 is coming from father, 23 is mother. Let's say white one is this came from father, this one yellow one is coming from mother. That's why they are homologous. They always stay mothers and fathers, different, like genetically, I mean, that coming from father and mother, and they are close, very structurally related, but not identical. So they make these pairs. Now here, homologous chromosome overlap. So what does it mean, the overlap? That means how can I draw like this? Maybe let's say one, and the other one is trying to always like just overlap like this, because they are structurally same. They can very well fit into each other. Because you see the previous picture, they have differences in the length. This one, you can see their length is different. Some are longer, some are shorter. So this chromosome's length is different, right? So likewise, we can see like this overlapping, right? So homologous chromosomes overlap. Right. How it overlap? So this is two homologous chromosomes. They try to reach each other. So the process of overlapping homologous chromosomes. So I can write like this. I write here process. You know, process is an event of overlapping homologous chromosome. Process of overlapping homologous chromosome is called synapses. So we give a special word for that one. The process of overlapping homologous chromosome is called synapses. Right? So then in this picture, they try to move, and this try to move. After that, it's overlap. The process is called synapses. And are they close with each other or they attached? So let's say if one, okay, use the color, then easy. Let's say yellow one And this is a white one, father and mother, homologous chromosome. Okay. So if this overlap mean they actually join with the protein molecules, they make this zipper-like structure. This is called synaptonemal complex. Right? Synaptonemal complex. They close with each other and they join to make the synaptonemal complex. So keep in mind, this is now prophase 1, right? Prophase 1, things are happening in prophase 1. Now remember what we learn in mitosis. What we learn in mitosis, we learn 5 changes. What are the 5 changes? Nucleolus disappear. Then the chromosomes become condensed and visible. Then spindles start to form. Then after that, we learn nuclear envelope disappear. And then a kinetochore try to attach the spindle with the chromosome. That 5 changes we learn. Now those 5 changes are also happening here. When you start, yes, chromosome visible and condensed. Nucleolus disappear. Nuclear envelope disappear. Spindle start to form. Right? And also at the end, spindle attach the chromosome. That's why I told, if you read mitosis and study meiosis only, you're going to learn these 2 changes at prophase 1 and metaphase 1. Keep in mind all these things happening. What is special happening is the overlapping of the homologous chromosome to make synaptonemal complex. Then when it forms the synaptonemal complex, they try to exchange the part of their DNA molecules. That means the sum of this DNA molecule is coming to this side, and some are going to the other side. White one is going to the yellow side. Yellow one is coming to the white one. So if a person is going to the other side, we call it crossover, right? We call it crossover. So here the crossover process. So synaptonemal complex is just like a kind of a bridge and allow molecules to come like going here and there. So part of the DNA molecules, they're going to other side and others coming to this side. All right, I think, yes, we will, I will WhatsApp update through the groups. We don't have much time. So I will continue from next time. Just any updates I will send to the WhatsApp group, right? So see you again next time. I just forgot the time actually. Okay, take care.
Thank you, sir.
Okay, bye.
Thank you, sir.
Okay, take care, bye.